So under the topic somite formation and neurulation, we have three parts. Number one is notochord formation. Number two is neural tube formation. And number three is somites formation and fate. As we know that this is the prochordal plate and this is the cloacal membrane, and the primitive streak, the cells of the primitive streak proliferates and move towards the prochordal plate. In the halfway, it forms a bulge, a bulge-like structure called the primitive knot or the primitive node. In the primitive node, after in a later point of time, there develops a small hole called the blastopore, and the cells pass through this hole in order to form the notochordal process. So now we are going to look into this in a transverse view, that is in this manner. So it will be like this. The same thing, we put it horizontally like this. So this blastopore is here. This is our primitive streak. This is our cloacal membrane here, and this is the prochordal plate. So we see near the prochordal plate, the mesoderm is absent here. The ectoderm and the endoderm are tightly bound, and the mesoderm is absent. Here also near the cloacal membrane end, there is no presence of intraembryonic mesoderm. So now what happens? This formation of blastopore, as we see here, after the formation of the blastopore, the cells pass on from here and they move inside in the mesoderm, in the intraembryonic mesoderm. But they cannot pass the prochordal plate. They are formed only till the prochordal plate, because here these two cells are joined. There is no space. There is no mesodermal space present here. This structure is called a notochordal canal. See, this blastopore, further the hole deepens and it is present all through here. Now after that, we are going to take a horizontal section of this. So in the horizontal section, we will see the epiblast cells and the hypoblast cells, and inside there will be mesoderm, right? Ectoderm and the endoderm and mesoderm. So just now we saw the transverse section. Now we are going to see the horizontal section. So in the horizontal section, it will appear like this. So this will be the notochord, because the notochord passes all over here. So at this point, when we take a horizontal section, it will be only like a small, this is the notochord, so it will be only like a small dot circular thing. So this is our notochordal process in the intraembryonic mesoderm. Then the cells of the notochordal process, after that, they form a notochordal canal. Here we saw when the whole of the blastopore goes inside till here, when the hole extends, it forms the notochordal process, forms notochordal canal. After that, the cells of the notochordal canal, they join with the cells of the endoderm. Then these cells, they disappear. The below cells, they kind of disappear and then forms like an arch, and again it forms like a plate. So this plate formed by the notochordal notochordal cells is called a notochordal plate. Then again it forms like an arc, and again it forms definitive notochord. So what happens here is, first it was like a mass of cells, then there was a canal, then there was like this, then the formation of notochordal plate, and finally the formation of definitive notochord. This is in the horizontal section. So we know that the cells that form the notochord migrate from the primitive knot by the formation of a hole called the blastopore. So now this migration occurs in two phases. In the first phase, the migration of the cells, when the cells are migrating, it only influences the formation of forebrain and the eye. It doesn't form, it only influences. When the cells start migrating, it influences the formation of forebrain and eye, and after it actually migrates, in the second phase, it forms the definitive notochord. However, the definitive notochord in human beings, only small remnants are present. Notochord usually disappears, and if it is still present, then the fate of the notochord will be nucleus pulposus in the intervertebral disc. It will be present like a structure called the nucleus pulposus in the intervertebral disc, but it does not give rise to the vertebral column. Now the second topic will be formation of the neural tube. So again we are taking horizontal section. In that we find above is the ectoderm, below is the endoderm, and in the mesoderm there is mesoderm as well as the notochord which was just formed. Then there forms a thickening in the cells of the ectoderm that is just above the notochord, this area. And at the edges of the thickening is called the neural crest, and this thickening is called the neural plate. So what happens is, this thickening deepens down, forming the neural groove, and this neural groove finally forms the structure called the neural tube, and the neural crest separates like this. This is the notochord which is gone little below from where it was, and this. So after the formation of the neural tube here, the cells in the mesoderm forms the paraaxial mesoderm. Then this is the intermediate mesoderm, and this is the lateral plate mesoderm. Paraaxial mesoderm, intermediate mesoderm, and the lateral plate mesoderm. Just because of the change in the shape of the structure of the mesoderm, it gets its name. Looking at the whole thing, we see that this is the notochord, this is the paraaxial mesoderm, then there is intermediate mesoderm and the lateral plate mesoderm here. It is marked. More clearer, now seeing the horizontal cut section, we see that this is the notochord, this will be the paraaxial mesoderm, this is the intermediate mesoderm, and this is the lateral plate mesoderm. Okay, so now the cells from the paraaxial mesoderm are the cells that are going to form the somites. So this part of the paraaxial mesoderm, just by the side of the notochord, forms the somites. So there are 44 pairs of somites here: 4 occipital, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 8 to 10 coccygeal. The somites form the axial skeleton and the muscles, the skeletal muscles. This picture is really clear, showing the prochordal plate, the cloacal membrane, the primitive streak, the primitive node, then the notochord. Here by the side of the notochord we have the somites formation from the paraaxial, then here we have the intermediate mesoderm, and this area is called the lateral plate mesoderm, the cells in this area. Cut section also showing here like this. The neural tube is all this area, this circular area. The notochord is only this area. Now moving on to the fate of the somites, we know that here was the neural tube, below it was the notochord. By the side, the paraxial mesoderm forms the somites. So this somites gets converted into three parts. The outermost is the dermatome, then the myotome, and the sclerotome. DMS. The cells of the sclerotome, which was on the most medial side, now migrate and they surround the neural tube. Then the cells of the dermatome, they migrate and they form the dermis of the skin. And the cells of the myotome, they give origin to the muscles. Okay, they still remain and they give origin to the muscles. They are divided into two parts. One is the epimere, which is innervated by the dorsal ramus, and the below part, the hypomere, which is innervated by the ventral ramus.